let's take a look at enzymes, what they are, and how they essentially work. And we'll take a look all of this as quick as possible. I know that some other videos have been quite long at 12 to 15 minutes, so let's try and make this a quick video. So for enzymes, let's take a look at its definitions, some of its basic functions with charts, of course, so what denaturing exactly is, and inhibition. So let's get a few definitions out of the way. An enzyme is nothing but a biological catalyst. What is a catalyst, you might ask? Well, a catalyst is nothing but an object that speeds up a chemical reaction. And it does this in two major ways. First, it increases the rate of reaction, or how many times the reaction is performed. And secondly, it decreases the activation energy. Activation energy. Activation energy is simply nothing but the amount of energy it takes to actually start the chemical reaction. And with talking about chemical reactions a ton, we have to look at what exactly is a chemical reaction in the first place. And a chemical reaction can be essentially summed down to making or breaking a bond. Whether it be from two molecules, two compounds, or just two elements, they all involve chemical reactions, and usually this happens in relation to electrons. So now that we got these definitions out of the way, let's take a look at its basic function, the basic function of enzymes. For that, we have to take a look at the structure of an enzyme, really. The basic enzymes are usually noted in something like this manner. This is the really basic drawing of an enzyme. And we can see this little cavity right here, and this is what we call the active site. What goes on in the active site? Well, essentially the entire reaction itself. Let's look at the reactants of an enzymatic chemical reaction. These are the reactants, and they're also known, and you should take note of this word, as substrates. Substrates are essentially the reactants in an enzymatic reaction, and there are two main ways that this enzymatic reaction can occur. The first way is what we see right here, is where these two independent substrates come and they form this product. A product of these two substrates linked together. Yet in another enzymatic reaction, and we had to draw the enzyme here again with the substrates will start as one compound itself, go into the enzyme, and come out as two individual products. So we can really sum down the basics of enzymatic reactions as this. Reactants, also known as substrates, go into the enzyme and come out as products in these two ways that we see here. But let's talk about really the structure of enzymes in relation to its environment. And what's important to know that enzymes are specific. What this means is that enzymes are really specific to the environment around them, whether it be by temperature or by acidity or pH. Enzymes are extremely specific to the environment around them. And this makes sense. As we know, enzymes are made of proteins. And what happens is that when the temperature gets too high, these proteins in the enzymes can denature. What this means is that the enzyme itself loses its shape. Why is this important? Well, much like how the enzyme is specific to its environment, an enzyme is also specific to substrates. Substrates are really specific as well. We talked about the active site, and the active site specifically binds to substrates. So if this active site changes shape due to this denature and this denaturization, then from all of a sudden, substrates can't enter the active site. Can't enter the active site. And we can take a look at graphs like this as well. If we look at a graph of, let's just say, the functionality, we'll call that function, in relationship to pH level, we can see that the enzyme will only work in a certain range. And I'll make this a different color. The enzyme will only work in a certain range. This is its peak functionality. Same if we look at temperature in relation to function. Temperature, the same goes as well. I'll put the enzyme in yellow this time to spice it up. It only works in a certain temperature range. Thing is, since enzymes are specific, the rate and their peak rate of reaction occurs in some different pH levels as well. For example, in your stomach, where the pH is really low and extremely acid, enzymes will work closer to the lower range of the pH scale. Yet, in some more basic parts of your body, it'll work at higher pH levels. And same thing goes for temperature. If there's a place in your body where it's really cold and there are enzymes functioning there, well, enzymes will work at the lower temperature range. Yet, and it goes the exact opposite in warmer places of your body. So once we look at the denaturing of enzymes in relation to their environment, it's also important to look at inhibition. Inhibition. And there are two main types of inhibition. Two main types. Inhibition is essentially where substrates cannot bind to the enzyme because of certain factors. These factors are, well, called inhibitors. Inhibitors. And looking at the two main types, the first one that we have is active site inhibition, also known as competitive inhibition. Competitive inhibition. And what basically happens here, if we draw again our enzyme, our basic drawing of enzyme, and the substrates, of course, is that this inhibitor, which we'll draw in red, actually blocks out the active site. It blocks out the active site so that now these substrates can't actually enter. Now, in the other case that we actually have non-competitive inhibition. Non-competitive inhibition. This is also known as allosteric inhibition. Allosteric inhibition. And this is actually a bit of a different case, and it's a bit interesting. So for this, we have to, of course, draw our enzyme again. And one important thing to note is that it isn't a perfect circle in these semicircular parts that I've drawn here. There's actually a bit of a groove right here on the other side. So what happens is that this inhibitor, which again we'll draw in red, doesn't bind to the active site, but actually wedges into these small gaps. And what it does is it basically denatures the protein, changes its shape to maybe something like this. And from what we know about substrates and them being very specific to the active site, they can't bind. They can't bind. And that's important to note when we look at the types of inhibition. So when you look at the inhibitors that are presented in many questions, when anything blocks the active site, it's competitive. If anything doesn't block the active site but still stops substrates from entering the enzyme, it's non-competitive. And then lastly, we told you we're going to draw some basic charts, and you'll get some of these questions in your pre-AP class as well. And for this, we have to look at the enzymatic activity in relation to the rate of reaction, and you'll get in a chart about the activation energy. So now, for many of these questions, you'll have to compare regular reactions to reactions with enzymes and point out which line on the chart actually correlates to a solution or an area with an enzyme in it, or with multiple enzymes. So what we note is that, let's just take a regular reaction without enzymes in purple, is that these have much different graphs, much different lines, I should say, on individual graphs. On this rate of reaction graph, it's actually quite low in relation to rate of reaction, because without enzymes, there's no way to speed up or increase the rate of reaction. Yet on activation energy, it takes more energy to start the reaction. That's something important to note. Let's draw the enzyme and the lines that correlate to enzymes in this case. The rate of reaction is going to skyrocket. because, well, that's the enzyme's function. Yet for activation energy, it's actually going to be lower because it takes less energy to actually start an enzymatic chemical reaction because, well, that's the enzyme's function again. So when you note these type of questions, always make sure to look at the y-axis and relationships between the y-axis and the rest of the graph. Because as you can see, in some cases, the enzyme and the line for the enzyme is much higher than the regular reaction. In other cases, it's much lower. So it all depends really on the y-axis and what we see there. So I hope that you've gotten a good understanding about the basics of enzymes. We talked about inhibition, denaturing, some basic broad concepts and vocabulary, and overall the basic functions of enzymes itself. So I hope you all have a wonderful day, and I hope that you've all put these concepts in mind.